Alright, we're doing an experiment today because I'm outside doing this lecture. So if you hear planes and dogs and all kinds of things, it'll add to the biology ambiance, I think. Okay, so an introduction. So the next two sections we're going to talk about kind of how cells are regulated as far as growth goes. So the introduction to this is talking about cancer cells. So cancer cells actually start out as normal body cells, and they undergo some mutations, which we'll talk about a little later on. They lose their ability to control their tempo and division, and they cause disease. So cancer therapy seeks to disrupt one or more of those steps in cell division. So in a healthy body, cell division allows for growth, replacement of damaged cells, and development from an embryo to an adult, which we'll talk about soon, but that isn't a really important thing to note. Okay? In sexually reproducing organisms, eggs and sperm result from mitosis and meiosis. We're just going to talk about mitosis today. Okay. So it looks sort of like this. We've got our cells, and maybe we've got a scanning electron micrograph, and we can see that they're beginning to pull apart from each other and become two new cells. So in this particular lecture, we're only going to look at cell division and reproduction, and we're going to look at eukaryotic cell cycle and mitosis. So these two things are what we'll look at today. It's a good plane, a big plane. I don't know if you can hear that one. Okay, so cell division and reproduction. So our essential question today will be, how do cells reproduce? And why is it important? Okay, we've kind of already looked at some of that answer, but that's the big thing. We want to look at how they reproduce and why it's important. So cell division plays many important roles in the lives of organisms. So the ability of organisms to reproduce their own kind is key to life. So above all else, reproduction has to happen to keep a species going. Okay, so that part isn't important. But we also need it at the cellular level just to kind of maintain a single individual. Okay, so cell division is a reproduction at the cellular level. Okay, it'll produce two daughter cells that are genetically identical to each other. This is key because we'll have some later that are not genetically identical. So it requires duplication of chromosomes. So we've got to copy the genetic material. So I'll give you that one as copying the genetic material. Okay, and the structures that contain the DNA. And then we've got to sort those sets of chromosomes into resulting pairs of daughter cells. So we've got to duplicate the chromosomes and then sort them. Okay, so living organisms reproduce by two methods. If we're going kind of larger scale here, we have asexual, and the prefix A just means not. So this is not sexual reproduction. So it produces offspring that are identical to the original cell or organism. And by, I don't know where that line came from, but identical to the original cell or organism is referring to genetics. So it's genes. So it involves inheritance of all genes from one parent. Okay, so all those genes are the same. They're sort of, you can think about it because it is sort of how it goes, is that they're technically clones of each other. And this actually happens in some animals. We can talk about that later on. Sexual reproduction involves producing offspring that are similar to the parents but show variation in traits. So it involves inheritance of a unique set of genes from two parents. So asexual is not sexual, so essentially one parent and the offspring are identical to the parent. And sexual reproduction has two parents that are involved, and then we see variation of traits among those. So cell division is used for reproduction of single-celled organisms. So things like bacteria are good examples of what use cellular division just as their normal reproduction. In multicellular organisms, however, it can be growth. So to get from a tiny baby to kind of a larger human like we all are now, it requires cell division. So repair and replacement of cells, so anytime you cut yourself or you scrape your leg or whatever, just anything that happens, if we need to repair damaged skin or if we need to repair damage inside due to some kind of bacteria, that requires repair and replacement of cells. Okay, and then also production of egg and sperm. So this will go into the genetics section, so we're not going to necessarily talk about it today, but it is involved in cell division. Okay, so this might be like a single-celled protist that's reproducing, kind of making a clone of itself. We also see some types of asexual reproduction in animals, for example, starfish, because they can reproduce or make more of themselves by kind of cutting off a series of limbs and then regrowing a whole organism from that. We'll get into that whole bit next semester, but that stuff's pretty cool. Okay, plants do this kind of thing all the time. You can do clippings, you can do, you know, take parts of like even potatoes and you can regrow them. That's all asexual reproduction. All right, but with sexual reproduction, we see a lot of variation among individuals, and that's the whole point. Okay, this is based like a ball of cells, so this is going to be kind of the mitosis thing happening here, so we can see that with this particular set of cells. This will definitely be mitosis, so we'll make more sense out of this kind of a diagram a little bit later. Okay, first, before we continue, prokaryotes for sure reproduce in a different style. It's going to be a lot like mitosis, but a little different because we're not dealing with the nucleus, obviously. So prokaryotes are single-cell bacteria and archaea, and they reproduce via binary fission. So fission means, you know, kind of dividing, and binary is like 1 and 2, so it literally means dividing in half. Okay. So the chromosome of a prokaryote is typically a single circular DNA molecule with associated proteins. And remember, they're a lot smaller than eukaryotes, so that's a good review. So binary fission of a prokaryote occurs in 3 stages. First, we duplicate the chromosome and separate those copies. Then we continue with elongation of the cell, movement of those copies, and then we divide into 2 daughter cells. It's going to look a lot like mitosis later, so think of these kinds of 3 steps. Duplicate chromosome and separate them, elongate and move those copies, and then divide into 2. That's it. Okay, so you can see that here we have a prokaryotic chromosome. So duplication of the sets of chromosomes, there would be 1 set here and 1 set here. Okay, it elongates, it continues to get longer, and we move those 2 copies into separate units. And then in the end, we split, and we end up with 2 daughter cells. Okay, so binary fission is fairly uncomplicated. Okay, so prokaryotic chromosomes will exist here in the middle, and this is them in the middle of division, where we have this happening. All right, so the next, I dropped my pen. There we go. Okay, so that's the basics of what's going on, and we're at 8 minutes, so it is a good time to take a break if you need one, because we're going to go into the eukaryotic cell and mitosis. Okay. You need to chunk your notes, write questions out to the side, write your summary, and then come back. So the eukaryotic cell cycle and mitosis are going to be really important. These are big things to know too. State of Texas loves this stuff. All right, so large complex chromosomes. So we got a lot going on here. So eukaryotic cells, remember, are more complex and larger than those prokaryotes. They're also going to have more genes, and they store their genes in multiple chromosomes within the nucleus. So we're more complex, we have more genes, and we store those genes in the nucleus, which is an extra step because we've got that whole nuclear envelope that might be in the way. So each eukaryotic species has a characteristic number of chromosomes in each cell nucleus. Ours is 23 as far as pairs go, so we could say with humans there's 23 pairs. Okay. So eukaryotic chromosomes are composed of chromatin. It's a long DNA molecule, and proteins that help maintain the chromosome structure and control the activities of genes. So basically a long DNA molecule and proteins. Chromatin really looks like spaghetti noodles. So it's what you look and you see that normal cell, and here's our nucleus, and then you see all this stuff in here. We can look at a cell model again if we need to, but that's chromatin. It just looks like spaghetti noodles in the nucleus. And so usually that's how I refer to chromatin, noodles. Okay. So to prepare for division, the chromatin has to become really, really compact. And we can actually see it in the microscope, and we will. So the next time that we do a little experiment, we'll go through and actually use the microscopes to see the cells dividing. But think about a bowl of spaghetti noodles. So this is what we have. If we were to try to divide this perfectly in half so we could get that into 2 cells, oh, it'd be a giant mess. So we really have to compact and kind of twist up our spaghetti noodles if we're going to get them into equal parts to make 2 separate cells. Okay, so that's the kind of thing to think about on that one. When they condense down, you'll start to see that we can actually see them, and they're not quite as noodly. They still are, but we're going from like angel hair to linguine, if you catch my drift. Okay. I like food. It's kind of obvious. All right. So what we'll have originally is like one single chromosome. Okay, so that's a compact version of chromatin. So the spaghetti noodle that's kind of condensed down is into a chromosome. And it's going to have one single DNA molecule in it that'll, remember it's our double helix shape. What we'll do is we'll take those 2 wound chromosomes, or that 1 wound chromosome structure, we'll make a copy of it and then actually sort of attach it together at what's called a centromere. The centromere is actually able to kind of keep track and keep those sister chromatids, or kind of exact copies, together until we're ready to move them apart into separate cells. Okay, so it looks kind of crazy, but we're going to talk about it. Okay, that's kind of where we're at. So a few more words here to this one. So before a eukaryotic cell can begin to divide, it has to duplicate all of its chromosomes. And it'll result in 2 copies called sister chromatids. They're called sister chromatids, but they are exact copies. Okay. All right. The sister chromatids are joined together along their lengths and are cinched especially tightly at a narrow waist called the centromere. It just helps them keep track, because we don't want to lose track of our exact copies. So when a cell divides, those sister chromatids will separate from each other and are then called chromosomes. Chromosome is like the functioning thing. So when we make a copy and then we split those sister chromatids apart, they become their very functioning own chromosomes. And we'll keep track of all those words. They are tricky. I sympathize with you on that one. And they sort into separate daughter cells. Okay. So we'll keep track of those. So again, chromosome, we make a copy. It's at the centromere that our sister chromatids are exact copies. Stick together. Okay, and then when we want to divide them into new cells, they'll distribute into 2 daughter cells, and then they sort of get called chromosomes at that point. But the big thing is that if you have 1 chromosome and you see that we're making copies and then we're going to split those copies between 2 cells, then you're half of the battle there. Okay, so the first thing that we need is we've got to talk about the cell cycle. So this is an ordered sequence of events that extends from a time a cell is first formed from a dividing parent until its own division. So it's basically the whole cell's little life cycle. So think kind of like life cycles, it's the cell cycle. It has 2 distinct phases that we'll talk about. The first one's called interphase. Inter means between, so it's the phase kind of between mitoses, if you want to go with that. Okay? But interphase is the between phase. This is actually where we'll end up duplicating the cell's contents. It has 3 phases, so G1, S, and G2. G stands for growth, so the first growth one increases cytoplasm. The S phase is for duplication of chromosomes. It's actually called synthesis phase, so we're making chromosomes. That's why it's the S phase. And the second one is growth. Again, so this is actual preparation for division. Then after that, we'll have the mitotic phase for mitosis, which will have division of the nucleus. That's what mitosis is dividing the nucleus, and then a separate thing called cytokinesis, which divides the cytoplasm. Okay, it's its own separate phase. Okay, next. So here's sort of what we get. Interphase is actually what you normally see the cell at. All the little cell drawings that we did, those are all interphase. But the thing is that if we want to get ready for cell division, we put it through interphase, the whole complete phase. So the first one is the first gap where we get growth. Okay, we get growth and we get cytoplasm. DNA synthesis is the S phase, so we copy our chromosomes. Okay, the next one is the second gap phase where we get a little bit more growth and preparation. Then we have the mitotic phase, which includes mitosis, a splitting of the nucleus, and cytokinesis, splitting of the cytoplasm. Okay, so we got those, and we'll kind of check through them in class and make sure we get them. But lots of diagrams to help us through this one. So mitosis progresses through a series of stages: prophase, prometaphase, metaphase, anaphase, and telophase. I will say it's not wrong if you see online where prometaphase is not included. That's fine. So most of the time we're going to see PMAT as the kind of way to do it. So prophase, metaphase, anaphase, telophase. For our kind of intents and purposes here, we're going to do, which sounds terrible, PPMAT. But whatever helps you remember it, right? Cytokinesis will often overlap with telophase, but it is its own distinct stage. Okay, so we'll go through those. First of all, mitotic spindle is required to divide the chromosomes. So it's sort of like if you've ever had to move something, it's kind of like a tow line. It's going to help kind of tow our chromosomes. Okay, so it's like a tow line or a rope, or a chain, whatever it is that you want to talk about that helps divide those chromosomes. So it requires separation of two sets of daughter chromosomes, or helps guide those. Okay. It's composed of microtubules, so it's those proteins of microtubules that really help that move along. It's just a series of proteins together. So spindle microtubules emerge from two centrosomes, which we learned are in the animal cells, that are microtubule organizing regions in the cytoplasm of the eukaryotic cell. Okay, so that's the thing that we're talking about there. We're going to get into these a little later. My videos never try to play while I'm doing my own video over top of them, but remind me because these are really good looking ones that we can take a look at. Okay, overall, here's our centrosomes, here's our chromatin, our spaghetti noodles, our nuclear envelope, and our cytoplasm, or our plasma membrane. We're going to go through each of these phases individually, but you can see some of the distinctions occurring. So let's go through them one by one, and we'll review all kinds of things. First of all, we've got interphase, which was that first diagram on the left, is that the cytoplasmic content doubles, two centrosomes form, and chromosomes duplicate in the nucleus during the S phase. So it's not during mitosis that we duplicate the chromosomes, it's during interphase. Okay, that's key. Okay, and so it just looks like your sort of normal cell. Sort of like that. Okay? Alright, there's your interphase. And now we'll talk about prophase and prometaphase. They're distinct ones that we need to know. So prophase, in the nucleus, chromosomes become more tightly coiled and folded. So we're starting to get more condensed, more seeable chromosomes. In the cytoplasm, the mitotic spindle begins to form and the microtubules will grow out of the centrosome. Sort of what that looks like when we saw that one picture. I'll come back to it here in just a second, but when you actually look at it in the microscope, you're going to see more dots and splotchy things in the nucleus. So it gets to be more kind of dotty and less spaghetti noodly. In prometaphase, the nuclear envelope will actually break down into fragments. Microtubules extend from the centrosomes. Some spindle microtubules attach to what's called kinetochores, which is just the parts where we're going to move the chromosomes. I can give you that one here. Parts where we move chromosomes. In case you need that. Chromosomes. Okay. Other microtubules will meet from the opposite poles. Okay. So you start to see that here. These are our centrosomes starting to form out, and you can see those chromosomes condensing further. Actually, let me go back just so you can see one more time. So you can see the centrosomes, the microtubules forming from those. You see the you see the two sister chromatids that are attached together and they're condensing down, and they're connected at the centromere. They get even more condensed down by prometaphase, and we actually have a kinetochore, which is just where the microtubules attach. Okay. All right, we're going to get into the second half. If you need a break, take one. You know what to do, but let's try to keep moving here. Okay. So metaphase, these are always fun to look at in the microscope. It's one of my favorites to look at, as well as anaphase. But in metaphase, meta sort of means middle. So this is where everybody lines up in the middle. All of our chromosomes line up in the middle. Anaphase, they will split from each other. Those sister chromatids are separated. And then in telophase and cytokinesis, we actually start to build two new cells. So the words to this, metaphase, is the mitotic spindle is fully formed, so that's all those microtubules coming out of the centrosome. Chromosomes will align at the cell equator. Kinetochores of sister chromatids are facing opposite poles. Okay, so they're all these guys are sort of lined up in the middle. That's sort of what's happening there. Everybody's lined up in the middle. In anaphase, the sister chromatids will separate from each other. Daughter chromosomes are moved to opposite poles, and motor proteins move along the spindle microtubules and the kinetochore short. It's just saying that we take the daughter chromosomes and we move them towards opposite poles. Like, that's perfectly fine to just go with that. Okay. Spindle microtubules are attached to chromosomes and they lengthen, moving the poles further apart. So it's just maybe elongating, like we said with the prokaryote. All right, at the end of anaphase, we'll have two ends of cells that are equal collections of chromosomes. Okay, so we're sort of seeing the divide happen here, and these are going to be equal sets of chromosomes, at least for the most part. Telophase is our last one. This is the cell continues to elongate, get longer. Our nuclear envelope will start to reform because we lost it earlier in kind of prometaphase. And then we're establishing daughter nuclei. The chromatin kind of uncoils, it loosens back up to get back to spaghetti noodle format, and then the spindle disappears. Okay, so they're pulling further and further apart and becoming two separate cells. You can sort of see that there. So during cytokinesis, the cytoplasm is actually divided into separate cells. Cytokinesis usually occurs simultaneously with telophase, but it is its own separate entity. In animal cells, cytokinesis occurs as what's called a cleavage furrow, which forms when a contracting ring of microfilaments interacting with myosin, and it deepens to separate the contents of two cells. What it looks like is like if you have one of those, I think of like, I don't know, like the Nike drawstring bags, how they've got the little things here, and you can pull right here, and you can pull each of these this way, and it actually shortens and closes up your bag. That's sort of going to be that contracting ring. I hope you can follow me on that one. So it's just like a thing sort of forms around this, and so each of these pulls, and it starts to pull in, so these will actually kind of move in closer together, and you actually break them into two. Okay. In plant cells, it's going to be a little bit different, though. We've got to form a cell plate, because remember, we've got this whole thing with a cell wall, and we actually need to rebuild the cell wall for making new cells. So we actually have little vesicles that contain cell wall material, and they form this cell plate. The cell plate grows outward to reach the edges, dividing the contents in two. Each cell now possesses a plasma membrane and a cell wall. So we'll move forward, but that's a good one. So here's a plant cell. If we're dividing, you can tell that we're going to rebuild that cell wall in the middle after we've divided the nuclei. So little vesicles come in with cell wall material, with maybe some cellulose and stuff like that. It starts to form a cell plate, and then it will work outward until it actually forms a brand new cell wall for our two daughter cells. All right, I moved awfully quickly through that, so do take your time. I'll see if I can put up some other little videos to kind of help you guys take a look at it. It can be a weird one, but we'll try to review and do some models as much as possible. The best thing you can do now is take notes about each phase and kind of get an idea of what's going on. Put all that stuff together, and then we'll come back and we'll review it together. So we have time for that. Okay? Again, as always, if you have any questions at all, shoot me an email.